Bismillah ar-Rahman ar-Rahim, paper 1-1, June 23, and this is the second video, question 22 to question 40. Question 22, the table shows some mRNA codons that code for a certain amino acids. A DNA template strand has the base sequence shown. So this is the DNA which they are showing you. Now we've got to make the mRNA from it. So I'm going to make the mRNA from it and then you can understand. What would be the change in the amino acid if the first base in the fifth DNA triplet? So okay, let's make the triplets. 1 ACA, GTA, TTA, TTT, GCA, and ACG. So these are the DNA triplets. So this is 1, this is 2, this is 3, this is 4, this is 5. So GCA, what would be the change if the first base of the fifth DNA triplet was substituted for an A base? So if it becomes ACA, now GCA, what would be the mRNA for that? CGU. What would be this for that? UGU. So CGU is here, is arginine, and UGU is here, so it's cysteine. So what would be the change in the amino acid if the first base in the fifth DNA triplet? First base in the fifth. So this is first, second, third, fourth. So this is the fifth DNA triplet, GCA. That's what I've written here. So what would be the change? Arginine to cysteine. Got it? Everybody please remember this is the mRNA codon. So this is the DNA. This is the DNA which they have given you. This got T in it, but this has to, you have to convert it. So from GCA it went to ACA because they said what would happen if the first base was substituted for an A base. Question number 23. Which structures contain cytoplasm with mitochondria and a nucleus? Xylem doesn't contain any. So the only thing is, we have to understand they were asking you three things: xylem, phloem sieve tube element, and companion cell. So the only thing which is present is A. Companion cells only contain all the three, you see. Xylem will have nothing in it. Phloem sieve tube element will have a little bit of cytoplasm, but it will not have a nucleus and mitochondrion. Question number 24. What is the correct term to describe intermolecular hydrogen bonding between water molecules? Water molecules along the xylem vessel, adhesion. With each other water molecules is cohesion. Now the diagram shows, question number 25, the diagram shows the outline of a xerophytic leaf that has been left for 45 minutes in different conditions P and Q. So one is a condition P and one is condition Q. Which statements about the cells in layer Y of the leaf in each of the conditions P and Q after 45 minutes are correct? So we're talking about what has happened after 45 minutes. There is less negative water potential in P than in Q. Yes, water would be more in P because water has entered, has become fully turgid. So less negative in P than in Q. That's correct. The cells may be turgid in P and plasmolysed in Q. That's also correct. So 1 and 2 is definitely correct. Now if you look at 1 and 2 is correct, so the only answer is A. There is no other in which 1 and 2 is correct. There is no net diffusion of water into Y in either P or Q. You say after 45 minutes, yes, there'll be no net. After 45 minutes, yes. It hasn't said what is happening in the 45 minutes. It says after 45 minutes. So there is no net diffusion of water into Y in either P or Q. So that is why the answer is 1, 2, and 4. Then question number 26. How does sucrose move from chloroplast? How does sucrose move from chloroplast to the phloem? Diffusion, apoplast pathway, or symplast pathway. So what we have to understand is we are talking of chloroplast to the phloem, right? So it has to be all three of them. Diffusion also occurs, apoplast pathway also occurs, and symplast pathway also occurs. So the answer is A. Because you have to understand is the chloroplasts are inside the cell in the palisade mesophyll or the spongy mesophyll, and then the sucrose has to diffuse out, and then it has to pass through the cells and the apoplast pathway and the symplast pathway, and then finally enter the phloem. Question number 27. How are companion cells involved in loading sucrose into phloem sieve tube elements? Now, either you know this or you don't know this. So the answer is A. Why? Because actively through co-transporter proteins and passively through the plasmodesmata. So this is the sieve tube and this is the companion cell. So the hydrogen ions pumped out and then the hydrogen ions move back in and along with them sucrose. And then you've got the plasmodesmata here. So the sucrose is going to move in passively through this. So actively through co-transporter proteins and passively through the plasmodesmata. So the answer is A. Then question number 28, the photomicrograph shows blood cells as seen using a high-powered light microscope. So we have 1, 2, and 3, and basically we've got to label them. So which row correctly identifies the different types of blood cells? Neutrophil, monocyte, lymphocyte. Either you know this or you don't know this. So the answer is C. What we have to understand is that one has to be the neutrophil, two is the monocyte, and three is the lymphocyte. Basically, the sizes are helping you to really figure this out. Question number 29. Plan diagrams of two blood vessels are shown. Which labels are correct? 1 and 2 and 3 and 4 and 5. Well, I would have sort of thought of it was what is 5? 5 is the endothelium. That was main would have been my way of figuring this question out. If you know it that blood vessels, the lining of the blood vessels is a single layered cell which is called endothelium. So that was my way of figuring it out because nowhere else is endothelium the answer. So that is why the question 29 is B is the answer. That's an easy way. If you know one, there's usually a catch in it. One of it is correct. If endothelium was written in 3 places, of course, then you would have to figure out the other, but endothelium is only written in 1 place. Question number 30. What are found in blood and tissue fluid? Carbon dioxide, yes, everywhere. Fatty acids, of course, would move out and in. White blood cells, yes, they can move out. Phagocytes can move out of capillaries. And proteins, yes, they can also. Some small proteins can move out. So the answer is A. 1, 2, 3, and 4. Question number 31. Which reactions will be slowed down by an inhibitor of carbonic anhydrase? Carbonic anhydrase catalyzes the reaction of carbon dioxide plus water to carbonic acid, H2CO3, and then the H2CO3, and the formation of, you see, H2CO3 breaks down into hydrogen ions and bicarbonate ions. So, question number 31, the answer is D, 2 and 3 only. Inhibitor of carbonic anhydrase. So carbonic anhydrase catalyzes the reaction of CO2 plus H2O giving you H2CO3, and then the H2CO3 breaks down into hydrogen ions and bicarbonate ions. So carbonic anhydrase inhibitor would result in 2 and 3 slowed down. Why would it be slowed down? Because if less H2CO3 is made, of course, less would be broken down into hydrogen ions and bicarbonate ions. You see, if normally 100 H2CO3 was being made and now only 50 is being made, so less of the 50 will convert into hydrogen ions and bicarbonate ions. Slowed down. Carbonic anhydrase basically catalyzes the formation of H2CO3, but if less H2CO3 is made, less of HCO3 and H positive ions would also be made. So that was a little bit of a trick question. Question number 32. Which sequence of letters correctly identifies order of events during the cardiac cycle? Either you know this or you don't know this. Very simple. Question 32. Which sequence of letter identifies? So Basically, the answer is C. Why is the answer C? Because T, atrial walls contract. So this is the first one, 1. Then V, wave of excitation enters the atrioventricular node. Then U, impulse is delayed by a fraction of a second. Then W, wave of excitation passes down the Purkinje tissue. Then Y, ventricles contract. And then X, wave of excitation spreads from the sinoatrial node. You can start it from X and then you can continue like that. So you can start it with X if you want to start with X. That could be the first one as well. So that makes it easy for you to then follow the sequence of events. Question number 33. A student of mine used to say कि मिस मैं ग्राफ देखता हूँ तो मुझे बुखार चढ़ जाता है. तो आपको बुखार तो नहीं चढ़ना चाहिए, but still खैर. The question you've got to understand is that you have to get familiar with this graph, and I usually use this line only. I find it very easy to use this line. I just use the aortic line. And I see, okay, the pressure, so here the semilunar valve is opening, and that's why the pressure is increasing, and then here the semilunar valve closes. So 2 and 3 semilunar valves, semilunar opens, 3 semilunar valve closes. Because aortic pressure rises only when the semilunar valve will open, blood enters the aorta, and then that semilunar valve has to close. So what we have to understand is valves open and close at the points numbered. Which row identifies the valves opening or closing at the points numbered? So 2 and 3 has to be the semilunar valves. So if 2 and 3 has to be the semilunar valve opens and then closes. So this is which row identifies the opening or closing. So I usually do it like this. So that is why the answer is D, because I have figured out that the aorta is the easiest for me to figure it out. Maybe so you use some other method, but for me, I find this graph a little confusing, and I like to use the aorta as the baseline. The aortic graph, the line of the aortic graph helps me to understand where the semilunar valve of the aorta will open and close. You could maybe do it. Some of you are very bright and you can do it some other way as well. I'm not saying my way is the correct way. My way is the highway. Question number 34. A person with no breathing conditions rests for an hour. Their breathing in this time is shallow and slow, so little air from outside the body reaches the alveoli. The person's heart rate remains constant. Which statement is correct? The carbon dioxide concentration in the blood in the pulmonary vein will be higher than in the pulmonary artery. Why? He's not exercising. Carbon dioxide molecules in the air of the alveoli move out of the blood by active transport. No, they move out by diffusion. See, the air in the alveoli has a lower concentration of oxygen than the blood in the pulmonary vein. The air in the alveoli has a lower concentration of oxygen than the blood in the pulmonary vein. Oxygen molecules diffuse from the air in the alveoli into the blood at a slower rate than when the person is active. Why? The answer is D. But why is the answer D? Because this person is at rest, but when he's exercising, what is going to happen? He's going to have lesser oxygen in the blood. So oxygen molecules will diffuse from the air at a very higher rate. Because if the person is very active, then he's producing more carbon dioxide and using more oxygen. So this was again a very trick question. And I'm sure majority would have got this wrong because you are not reading it very correctly. Question number 35. Which statements about all bronchioles are correct? So they would have an epithelium and they would have muscle tissue. They all do not have goblet cells. So that is why 35B is the answer. All bronchioles. You see, you're talking of which bronchioles? You're talking of the trachea divides into right and left bronchos, then terminal bronchiole, respiratory bronchiole, and the alveolar duct. So all do not have goblet cells. Then question number 36. The diagram shows some of the pathogens that cause disease in humans and some of the ways which are transmitted. What is the correct pathogen and method of transmission for malaria? Malaria, the pathogen is a protoctist. It's not a bacteria, it's not a virus, it's a protoctist, plasmodium. And how do you get it? You get it by a mosquito bite. Bite is a wrong word, but I don't agree with that. But still, I can't disagree with Cambridge. So question number 36, the answer is C, 2 and X. Then some of the processes which result in the formation of a population of bacteria that are resistant to a new antibiotic are listed. So you have these you have these bacteria, and you tell the patient to take antibiotics for 5 days, and then when he takes it for one day, he feels better, and he stops taking the antibiotic. Well, most of them die, but some are left, and those ones which are left, they mutate, and when they mutate, then now they are dividing, so their numbers are going to increase. So what is the correct order for these processes? The random mutation is the first one. The random mutation has to occur for the resistant bacteria to suddenly increase in numbers. So that is why the answer to question number 37 is D. So they have to mutate. The mutation has to be first occur, and then of course their numbers, because it all divides by binary fission. Once one bacteria is mutated, then all the offsprings from that bacteria will all be mutated. So the mutation is the first thing. So 4 would be the first thing to happen, random mutation. Then increase in genetic variation within, then increase in change in reproductive success, and then increase in frequency of the resistant allele in the population. Question number 38. When bacteria grow in a petri dish containing discs with antibiotics, there will be zones of inhibition of bacterial growth. So that the clear area means that the antibiotic has diffused out and it has killed all the bacteria. So this area, the bacteria are still growing. But this area, the bacteria have died here. So this is the clear zone of inhibition, and the antibiotic has come out of here and killed all the bacteria. The chart shows the sizes of the zones of inhibition when a species of bacteria was incubated on 5 different plates of agar, each containing a disc with a different antibiotic. So you see the zone of inhibition is 15, whatever, 17 millimeter. Then the zone of inhibition here is 4 millimeter. Then here it is 22 millimeter. And then here it is 19 millimeter. And here it is 10, 11, 12 millimeter. Which conclusions can be made about the most and least effective? Well, this is the least effective, and this is the most effective. So least and most effective would be 3 and 2. So that's very easy. If you know this, what is the zone of inhibition mean, only then you can do this. So that is why the answer is A. Most effective 3 and least effective 2. Question 39. Which blood cell type does not recognize, engulf and digest non-self particles? It is D. T killer cells cannot do that. Macrophages do that. Neutrophils do that. Phagocytes do that. Question number 40. Repeated infections with malaria result in more effective immunity to malaria. Which type of immunity is responsible for the most effective immunity? Naturally, if the person is suffering from malaria and he is getting better and then again suffering and then again getting better, so it has to be natural active immunity. Why? Because activity is done by his lymphocytes. He suffers from malaria, then he gets better, then he suffers, and then he gets better. So is responsible is natural active immunity. So the answer is C.